piece of paper and put that piece of paper over our source of illumination. Look in here and see if we can find that edge. So you may have to move that piece of paper. Typically, if you're halfway over that source of light, you're gonna be able to see the edge that is present. Right now, this one's not very in focus. So I'm going to move my condenser until that edge becomes nice and in focus. You'll notice that as I go under focus, it turns a little blue. As I go over focus, it turns a little bit red or yellow, orange. So you want to be right in between the reddish orange color and the blue. So now I've focused my light on my sample. I have my eyes focused on that sample. So I've got everything in here adjusted for the 10x lens. The next thing I want to do is notice with which of my eyepieces is not focusable. So I'm going to close the eye that is on the eyepiece that is focusable and use my fine focus to focus on something, whatever I choose it to be, and I want to get that just perfectly in focus, and I want to memorize where I have that. What does it look like being perfectly in focus? So I've got that piece. Now I close this eye, but I'm going to use this focusing mechanism on the eyepiece to get this eye in precisely the same plane of focus as that eye. So that's why I want to be maybe back and forth, check this eye and see what that little piece looks like. Go back over here and check and make sure I've got it in the same place with this eye. So now when I'm looking through here, both eyes are in the same plane of focus. My eyes are not constantly fighting to get things into focus, and you will not have problems with headaches when you're looking through the microscope. So I've got that all set for the 10x lens. So now I want to dial in. You could repeat all of this for the 20x lens, but typically I'm just going to go right on up to the 40x. Now notice as I go from that 4x objective, lots of working space here. Don't have to worry about it. Now you get that 10x, not as much working space there. When I get to that 20x, well, not as much, but not really in any danger of running into our sample. But when you dial in that 40x, that's getting a little scary. There's not much working distance there, which is why it's really important to have the parfocal objectives, because you're getting very close. The working distance isn't very big here, and if you haven't got things in focus, you could smash your 40x lens into your sample and break your 40x lens, which of course is the most expensive lens on this particular microscope. So don't worry too much. If you've got everything else in focus, fine, just dial the 40x lens in. You're not probably going to hit. We want to then repeat. We're going to focus, fine focus. We're going to then put our piece of paper on our illumination and again adjust my condenser so it's right between that shift between red and blue, sharpest focus, so we've got the best illumination. We have maximized resolution. We're going to be able to see things just as best as they can. Maximum resolution. Quite often what you'll find is as you go from the 4 to the 10 to the 20 to the 40, you're going to have to up the brightness a little bit because you're seeing less and less and less of the field of view. It's inexpensive to get a big lens with big viewing area when you're at 4x magnification. As you get to 10, it's a little more expensive to make that lens, so it tends to get smaller. At 20, a little bit smaller. 40, a lot smaller. So we're just seeing less of the field. Everything's magnified more. All right, last thing that we need to do is adjust our iris diaphragm. So here on your condenser, you'll notice a slide that goes from almost no light coming through, completely shadowed, all the way up to full light coming through. And what you want to do, looking through here, make sure you're focused. Start closing down your iris diaphragm, and when you're looking through here, you'll be going, gee, there's really not much in my sample. But as you start to shadow, oh my gosh, there's a lot of stuff. There's a lot of bacteria in here. There's a lot of small little things. I could not see them at all when I wasn't shadowing. So how much do you have to shadow? It depends on your eyes, depends on the lighting, how fine focus you are, exactly how much stuff is in here. So you might want to constantly be playing with that shadowing. So you're seeing the maximum amount of material. It's not getting too dim. You're seeing a lot of the organisms. So there's lots of bacteria. There's some flagellates in here. We've got some hyphae. So we want to be able to resolve all of those different kinds of organisms that are in our sample. If we have a camera, we can take the cover off of our trinocular port and just put your camera right in there. And we're all ready to start.